This is episode 22 of the Immunology Podcast, Maternal Immune Cells, with Dr. Francesco Colucci. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Roud. Welcome back to the Immunology Podcast, where we have conversations with immunologists. Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. Today we have Dr. Francesco Colucci from Cambridge University on the podcast to talk about his research on the role of uterine immune cells in pregnancy. We've also got our usual roundup of recent highlights in immunology news coming up. But first,
explore scientific resources for your immunology research at the STEMCELL Technologies Immunology Learning Center. Choose from different research areas and find expert interviews, technical tips, educational webinars, instructional videos, and much more. Visit stemcell.com/immunology-research.
well, Brenda, you just came back from Argentina, didn't you?
I did, just freshly out of the plane.
Well, the
No
good
jet
news
lag.
is you don't sound like a robot today on this podcast.
I know. I'm sorry, listeners, for last time's really poor audio, but you'd be surprised that it looks like good internet has not arrived into the Argentinian Andes. My apologies.
Alas. Well, while you were out, we got some hot Twitter action going on from openboxscience.org has been starting to tweet and see what we're doing. They're a new collaboration here that is an NGO that helps set up free and virtual talks for scientists. I don't know if you've been able to check that out.
I noticed. Thank you guys so much for the exposure. It was really nice to have some likes from them. So my shout out to Juan Huang, Jerry Lean, and Eugenio Contreras, who are the people behind the enterprise.
Brendan, I'm just going to have you say the names of everyone that comes on the podcast because, your 7 languages you speak or whatever makes life much easier.
Especially the Hispanic ones. Those are my forte.
I got Spanish okay, but you have all the other ones.
Well,
Dutch,
thank you.
French,
It's just a gift.
German.
It's a gift. Merci. But I'm glad to be back and just to talk about a couple of really cool papers I stumbled across. And also hear about what you have today.
All right. Well, why don't you go first? You had so much time to read on that plane. I'm sure you are ready to go.
I know. Okay. I'm going to just talk. I have to say, I'm very excited about these 2 papers I chose. They have been creating a little bit of, talk around the science Twitter. So I thought it was really cool to talk about. These 2 papers are talking about the link between Epstein-Barr virus infection and the development of multiple sclerosis.
Oh. So the papers have gone viral, you're saying?
Yeah, yes. Wow, what a pun. I have to say, I was not expecting such
bad jokes, but for all the miles.
That is your forte, bad jokes. Exactly. So these viral papers about viruses and autoimmune disorders have been a really great read. And it's nice because they really complement each other. So I'm going to start with the first paper that was published. It was already a couple of weeks ago, so it's not the freshest paper in the blog, but I just really wanted to talk about it. So this paper was published in Science by a group at Harvard. First authors Kjetil Bjørnevik and Mariana Cortese from the lab of Cassandra Munger and Alberto Askerio at Harvard, as I said. And it was published in Science, and it's called Longitudinal Analysis Reveals High Prevalence of Epstein-Barr Virus Associated with Multiple Sclerosis. So it has been known for a while, or has been seen for a while, that there seems to be a link between Epstein-Barr virus infection, or mononucleosis, as other people might know it, which is very common. So most adults, like 95% of adults, are seropositive for this virus, so have been infected with it at some point in their life. So it's extremely ubiquitous, and it has been there has already been a thought, or there's a lot of evidence that there's just a link between infection with this virus and the development of multiple sclerosis. It is always very hard to establish causality with this kind of incidence numbers and the fact that MS is very rare. So as you cannot really do a kind of a clinical trial to see if EBV infection will give you multiple sclerosis, this paper capitalized on a source of information that I wouldn't have thought about it, but it's brilliant. They followed 10 million subjects from the US Army. So they follow people that got into the US Army, and they had to give serum or like blood samples at that time for HIV testing, and they were tested regularly throughout their service. And they followed the patients that were initially EBV negative. So they were seronegative for EBV. And they follow them throughout the time. And from these, some of them developed MS. So what are the numbers? 10 million subjects throughout this study, which lasted 20 years. Around 955 people developed multiple sclerosis. From this 955, 35 were initially EBV negative. And so what they show is that throughout the follow-up, these people that developed MS, an EBV infection was always right previous to the development of multiple sclerosis. It was always preceded by a seropositive result, except on one patient from the 35. And this seroconversion which was a 97% seroconversion, was substantially higher than in patients that did not develop MS, which was about 57%. So these numbers add up to around 32-fold increased risk of MS after EBV infection. They compare this to another virus that is transmitted similarly, which is a CNV, and they show that CNV infection was no increased risk of MS with after CNV infection, which would to some extent control for lifestyle or other kind of exposures, because these two viruses have very similar epidemiological presentations. So they started with this, and then they also show they used a measurement which is neurofilament light chain concentrations in the serum, which is a fairly kind of general marker of neuroactional degeneration, which is not really specific for MS, but they also show that this neurodegeneration is increased after EBV seroconversion in the patients that developed MS. They also, so what they try to find is kind of try a little bit more take a closer look in the relationship between these two events, and they show that within the antibodies that are raised in these patients, many of them are, so they have a lot of them are specific against EBV, and that so they also see that these patients are responding against EBV. And what they say, so they also they make a connection between B cells and the development of MS, also because they also mention the fact that one of the most effective treatments for MS, it's not really clear how, but it is actually anti-CD20 monoclonal antibodies, which are depleting memory B cells or B cells mostly in the blood. They're not as good to go through into this cerebral spinal fluid, but they show that so with this information they put together that it is the immune response against EBV that somehow is triggering MS, the development of multiple sclerosis. And I thought it was, so they provide really strong epidemiological information or kind of evidence, and they show that they find these antibodies that are against EBV, and they made a really good case when it comes to kind of the epidemiological numbers. And this is a really nice connection to the next paper that came up a couple of days later, or maybe a week or so after, in which they actually look at a mechanism by which EBV can trigger this immune response that is related to B cell responses in MS patients. So then maybe we can talk about your paper, and then I can come back.
Whew. So real quick there before we shift gears completely and then come back. This kind of reminds me of what happens with strep throat and rheumatic fever, where you get like this response to strep, and then sometimes it attacks the heart valves, and some subset of kids it's not treated well, or even like celiac disease, right? Where like some people develop an autoimmune antibody. So it sounds like once in a great while, there is this terrible accident and it cross reacts. Is that basically what we're getting at here?
Yes, and that is exactly what the next paper goes around.
All right. Well, I will not steal your thunder
Yes.
then.
So
All right,
just
so
just hold your guns because I think it's a brilliant story. Like both of them together coming at the same time. It was a really nice
some
read.
editors totally held this back or talked to each other to have them come.
Yeah, but it's funny because this came in Science, and when I want to talk, the next paper I'm going to talk about came in Nature.
Yeah, they
or
talk
it's
to each
it's
other.
coming.
I mean, this is a thing, right? Like when the genome was produced, you're like, all right, who gets which one? Let's do it. No team.
Yeah, well, they all want a piece of this. So
Exactly.
what cool story do you have for
All
me
right,
today?
so I went to some nano engineering here, lipid decoys. So this is immuno-engineered nano decoys from the multi-target anti-inflammatory treatment of autoimmune diseases. This paper is actually in Advanced Materials. First author is Mengying Ho, and it was published super recently in January 19th. So this paper does some really interesting stuff. It takes lipid nanoparticles, or not lipid nanoparticles to start with. It's a polylactoglycolic acid nanoparticle, and it can be a disc or a sphere. And they take it and then they dip that in lipids from macrophages, raw cells, murine macrophages, before or after pretreatment with interferon gamma. And if they pretreat with interferon gamma, it activates the cells more PD-1 ligand, more PD-1 L expressing macrophage membranes appear. So those macrophages make more PDL1 and a bunch of other things, and show that by pre-treating the macrophages, it enhances the properties I'm about to talk about. So what you do with these fancy these fancy membrane-coated nanoparticles made out of a polyglycolic acid, well, you can use it to scavenge cytokines and PD-1L inhibit ligand PD-1 that's floating, that is actually immune activating instead of immunosuppressive, right? Because it has the opposite role if it's the soluble form versus the membrane bound. So what they do is they show both in a rheumatoid model and in colitis, and then more generally, like if you take this and put this in cell culture and look at what's in the media that's been absorbed by these, or any other system, or in the circulation, and they do kinetics in the blood circulation as well. But they show at the very highest level of this, you inject this, it soaks up cytokines, it soaks up soluble PD-1, and decreases inflammation and disease outcomes in a rheumatoid arthritis model in the mouse, and in the DSS colitis model. And they show that the pre-treatment is really important for the extra functionality. And they basically serve as sponges, right? Because they have all these receptors that can like suck up all the cytokines and absorb things, but that they can't do anything, right? Because they're not a cell that can then signal when their receptors are bound to something. So it's very, very interesting. I was very interested in like this kind of concept of nanotechnology here that they're using. Because then you could also reproduce it pretty well, right? You can take a standardized cell culture, standardized treatment, fractionate it. You would have to switch to human cells. But yeah, they do the ZIA model. This is I have to pull up the exact acronym here. This is one of the standard models of RA that they do this in. And then they do DSS colitis, but they show that not just like, oh, cytokines are absorbed from the bloodstream. It downstream affects the disease status, and that's what I found really interesting. So it's a general application, but you could then think about tuning it for different cell types that you'd coat with to then get different properties or different absorption. So I thought it was just a unique, different type of treatment. We've had CAR T cell therapies. We've had, I work on microbiome therapies. You have immune checkpoint inhibitors, but what about like good sponges? We have Enbrel, which is a pseudoreceptor, but that's one protein, right? And they show this outcompetes anti-TNF inhibitors and outcompetes other systems that are just single molecule targets. It's polymolecular, right? So it can absorb multiple different cytokines. and
That's
then
pretty cool.
pull it in. Yeah, and so the
But
paper really goes step by step, shows it in vitro how it works, shows that you have better properties with this pre-treatment, goes on, shows that if you just have a mouse in general, what it absorbs, inflamed mouse, they do live imaging, they then go for disease model after disease model, and then really hit the 2 disease models. They do biodistribution studies as well.
so the specificity, they coat these nanoparticles to give them the specificity against the molecule they want to absorb.
Right. They take raw macrophages, raw 264.7. They take those macrophages, pre-treat them with interferon gamma to upregulate some of the receptors to make them better. And then they like sonicate it and break the membrane up, throw the particle in there that coats the particle.
Right.
And that makes the drug. You guys can't hear the air quotes, but there's air quotes there. Can't see it. But that makes the drug, right? That we're talking about. And then they inject that, and then, or they can put it in culture and show effects there, but they can inject it into a diseased mouse, and it ameliorates the disease by absorbing the cytokines.
That's super cool. And is there these particles? Do they get taken out? Like how do they get degraded?
They get cleared over
cleared
time,
out.
right? So they're lipid particles. So they're gonna. I don't think they did excretion in liver versus kidney, but they're gonna get degraded over time.
Okay, so they won't accumulate or generate any other
they'll fall
effects.
apart as far as they can see.
I agree that's a really cool what do you say, approach. I never thought about putting sponges to take out this cytokines.
Yeah, well, there's
this
one drug
molecule.
that does it. As I said, Enbrel's a pseudoreceptor for TNF, but this works even better than Remicade or Humira, and a monoclonal antibody version,
Yes.
which is stronger because it has killing effects. Like anti-TNF antibodies kill cells that have too much TNF on them, which makes them work better than Enbrel soaking up. But this doesn't soak up one; it soaks up a bunch, and that's what's so neat. It's not just one molecule, right? It's the whole cascade. If you take an activated macrophage, it acts like an activated macrophage sink. So instead of signals going to the macrophages, it goes to this decoy.
Very cool.
It's really neat. So I liked it because I like biophysics.
Well, thanks for sharing, Jason.
All right, so tell me more about MS and how the immune priming and the accidental cross-reactivation.
Well, gladly. So after reading this first paper that was published a bit before, I thought, wow, that's pretty cool. And then sometime later, a new paper came up. And this is kind of presented ahead of this accelerated review from Nature. So it's technically not still published, but it's there available. First author Tobias Lance from the lab of William Robinson at Stanford. And in this case, the paper is titled Clonally Expanded B Cells in Multiple Sclerosis Bind EBV, EBNA1, and Glial CAM. So this already gives you an idea. So they find a really nice mechanism, one of the mechanisms by which EBV could trigger MS, through finding molecular mimicry between a protein from MF, a transcription factor from EBV, and a protein that's expressed in the CNS, which is known as glial CAM. So let me walk you through this. What they did is they took cerebrospinal fluid from MS patients, and they looked into the repertoire of antibodies that are expressed, of the B cells that are expressing antibodies in the context of the CSF. So what do we know is that that there's a big B cell component in the pathology of MS. There's some certain characteristics of MS that point towards the importance of B cells in this disease, such as the expression of what are known as oligoclonal bands, which are specific clonal antibodies that are shown as distinct bands in a gel when you look into the CSF of MS patients. And as I mentioned before, the fact that if the depletion of B cells is apparently one of the most efficacious therapies for MS, although it's not kind of good enough for being a real therapy, but it does reduce the incidence of MS in patients that are relapsing. And as I mentioned, and following from the last publication, there is a strong correlation between MS and EBV infection. And there's a lot. There are often found antibodies against EBVs. EBV antigens are found in MS patients, and there's a certain kind of hotspots, certain in the sequence of antigens from EBV, such as EBNA1, which are particularly found to be immunoreactive in these patients. So they took CSF and blood samples from 9 MS patients, some of them which were at the onset of disease and some were having an acute relapse. And they isolated the B cells from these cells, from these fluids. They also noticed, for example, that there are higher plasmablast counts in CSF of MS patients. So there's a lot of plasma cells that are producing antibodies there. And there is a high abundance of basically IgGs, which suggests that these B cells have matured and are producing they're producing a lot of antibodies in there. And they do a lot of work by single cell sequencing all of the heavy full length paired heavy chain and light chain sequencing of the plasmablasts in CSF and blood of these patients. And they also include some control patients, which are patients that have other type of neuroinflammatory diseases as controls. And they characterize this repertoire, and they already notice really stark differences between MS patients and other inflammatory disease patients, and between the CSF and the blood. So they show, and this was already kind of known, that there is more oligoclonal B cells in the CSF, and compared to the blood. And this is also the case for patients with MS versus other neuroinflammatory diseases. They show that there's these antibodies or these receptors have evidence of somatic hypermutation. So these have been really matured antibodies inside the CSF. And what is also interesting, there's a skewage towards 5 particular heavy chain variable genes, which is also kind of had already been described. So far, kind of this is, I think, was mostly expected. But then what I think is really, really cool is that they look into all of these. They take 148 BCR sequences and they express them as monoclonal antibodies. And then they use a protein microarray that contains lysates from EBV and also proteins and peptides that are kind of encompassing all of the EBV proteome, and also lysates from other viruses, particularly all other viruses that are neurotropic, so CMV and the like. So other herpes viruses. And they show that 1/3 of the express of these highly monoclonal antibodies that they express as monoclonal antibodies derived from the CSF fluid are binding to EBV proteins, and about 20% are binding to other viruses that are also found that are also associated to the CNS. What is interesting is that 8 of 9 patients had monoclonal antibodies that bind to the EBVNA1, which is a transcription factor from EBV. And they kind of really focus on one particular antibody, which they call MS39P2W174. I'm just going to mention it to MS39 monoclonal antibody, which binds to a particular region that it has been associated with with MS associated antibodies, which is 365 to 405. And it's really nice. They really show how this antibody is binding. They do some crystallographic analysis. But what is really cool is that they find that this monoclonal antibody not only binds to EBVNA1, but it also binds to glial CAM, which is this protein that is expressed on mostly astrocytes and oligodendrocytes in the CNS. They do this by using a HuProt microarray. So basically, a microarray is spanning 80% of the human proteome, and that's how they found this binding. And when they compare the binding between EBBNA1 and glial cam, there's some differences which explain why this antibody probably kind of came up in the first place. This antibody is hypermutated, so it's different from the germline. But the germline receptor can also bind to EBBNA1, so which already naive cells already have a binding predisposition to this particular antigen, and it is only the hypermutated antibody that will bind to glial cam, and more favorably, the binding is lower than for EBBNA1, but it is favored by particular phosphorylations. There's two serines in the sequence of glial cam that increase the affinity of this antibody for the protein. So what the authors suggest, and I think makes a lot of sense, is that these B cells started up binding and being activated by recognition of EBBNA1 and suffer hypermutation, and that hypermutation allowed them to also bind to glial cam. And this is where you have the molecular mimicry in which the antibodies cannot bind to the endogenous protein and the EBV derived one. And they test this also they look into targeting of glial cam in other patients. So they have serum samples from other MS patients, and they show that it is quite frequently they find antibodies that are binding to this particular epitope of glial cam, and also higher when it is phosphorylated. So what this paper shows, and I think was really cool, I thought, is that they show that this particular target in MS patients, if you take PBMCs from MS patients and you activate and you stimulate them with EBBNA1 or glial cam, some patients have actually very strong CD8 responses against EBBNA1, both in healthy MS patients, but mostly in the MS patients that this response also cross reacts with glial cam. So there seems to be really a kind of redirection of the immune response against this antigen that is very similar structurally to an EBV-derived peptide or sequence.
So what next? So do you try to
vaccinate against EBV? I don't know. I think.
Will the vaccine cause MS?
Well, I guess.
Or do you pick like a completely different epitope that's not that one, so it can't cross-react? And how do you map that? Like maybe that's it. Maybe you have to now figure out the part of the EBV that's generating this antigenic complex, which I presumably can know, so you don't do that one.
Yeah, I think that's it. The problem is that, and they show now, I was also doing some research to understand the context around these papers, is that there's a lot of like different peptides. It's not this only peptide. There have been other peptides derived from EBV that have been associated with MS. So I don't think the EBBNA1 is a nuclear. Sorry, it's not a kind of a surface peptide. It's a transcription factor. So maybe it wouldn't be expressed. So maybe you don't need that one. You want to go with something that is expressed on the surface of the virus to have prevent infection. But I think that you need to, mostly MS or you can get ABV infection without any kind of not really noticing, without a severe acute infection. So right. I think it is the acute infection that really gets the virus, gets the immune system kind of wired up to.
So maybe if we build the right vaccines, we'll be okay.
Yeah. I heard somewhere that Moderna was developing, but how do you test it? How do you do clinical trials? Like, you have to get super young children, because you get adults, makes no sense to vaccinate adults against it, does it?
Yeah.
So I mean, it really feel like I wanna. If I ever have a kid, I wanna get it vaccinated. But how? How do we get this vaccine out?
I agree. All right. Well, we've been deep diving for a while here, so I'll make this last one quick. This one is The Immunoregulatory Landscape of Human Tuberculosis Granulomas by Aaron F. McCaffrey et al. Last author is Michael Angelo in Nature Immunology, that just came out the 20th of January. So this is an interesting paper in that it takes old records of granulomas, old tissues of tuberculosis granulomas, and tries to really understand the signaling in them, because they're so rare to get and try to process with tuberculosis infection and catching all of it. And we've kind of eradicated most of it. It's a hard disease to study, but it's still a big problem. So what they try to understand is what the microenvironment of a granuloma is. And what they do is they use this very interesting technique called multiplexed ion beam imaging by time of flight, MIBI-TOF. So what they do is they fire an ion at the tissue section. First they coat the tissue with 30 some odd antibodies. I want to say it's 37 with different metals attached to them, right? So, mass spec style antibodies, and basically do an IHC, but you can't make that many colors up, right? But it's same concept for different immune complexes and cytokines, and so different cytokines, different cell surface markers. So super duper flow, but it's an IHC, right? And then they blast the whole section, but they know positionally where they were blasting at to suck off that little small spot. And then they send that small spot through a mass spectrometer to then measure the molecular weight that went in through there and convert that molecular weight based on what was there to, it was a CD4, because that's what we said, this metal ion attached to the antibody was. And so they use that to rebuild a histological image of the whole thing with 38 color, so to speak, if you want to think about it that way. So like a 37 air quote color IHC with pretty good resolution, right? And then they use that to understand what's happening in the granuloma. And they do it all the way through. They do a whole bunch of modeling and measure spatial relationships with a system. They have another acronym for this that I cannot remember. I'll give it to you in a second. It's where is it? Spatial LDA is what they call it. Latent Dirichlet allocation. Especially, it's like when we think of TISNE or UMAP, but for spatial relationships to each other a little bit differently. So they were trying to finger single cell identity in granuloma structure and how they spatially relate. And they go through all of this, and there's 27 supplemental figures. And they get to the idea that in granulomas, in TB, interferon is depleted and TGF beta is elevated, and it's enriched in regulatory C cells and IDO1 and PDL1, which are both in immunosuppressive signaling pathway myeloid cells. And the blood of people with TB mirrors these trends. You can find MAR IDO1 and PDL1 as well. So you can see a signal in blood. And PDL1 expression is associated with progression to active TB and treatment response. But if you look at other granulomas, not the whole story is not true. I think they went and looked at sarcoidosis as a model here. And in sarcoid, they were able to find PDL1 was high, but the IDO1 was not. And so it really shows that they were able to figure out that the granuloma is a specific environment, identify PDL1 and IDO1 as systemic markers, which is really interesting, but really then showed at a high level that it's enriched in TGF beta and down in interferon gamma. So what does that all mean? I think this hits nature level because of the technology used to be able to basically scan old slides point by point with 37 antibodies and figure it out. It's a hard paper to read, particularly because they don't go into the technology a lot and you have to really dig. And there's 27 supplemental figures, which is a lot of supplemental figures. It just keeps going. And it's not like one panel supplemental figures. It's like a lot.
Well, some scientists are very thorough with their supplementary information.
Yes.
So, but just to be clear, these IDO and PDL1 is expressed basically on the myeloid cells in these granulomas.
Correct. And there's more Tregs in there too that they're able to find.
Right. And this is something that is specific of MTB derived kind of granulomas. Probably right. Is the MTB is doing something, is helping this to generate this environment.
Yep.
I think that was already, I don't want to say already known, but I think was already associated that maybe, I don't know, I'm not exactly know about.
The IoT wasn't. The interferon TGF beta was, but they weren't able to dive into the granuloma itself and figure out what was happening there.
Right.
They're able to do this segmental sectioning fanciness. And I think that's really what the paper's kind of impact is about, is that.
Yeah, I guess that's very important because the granuloma is so important, the structure of the thing is so important to understand the relationship between the cells in the granuloma that having this really spatial resolution makes a huge difference.
Exactly.
All right. So a shout out to the authors. I'm just also checking out the paper, looking at the figures, and they're so nice.
Yeah. All right. We're going to be talking to Dr. Francesco Colucci at Cambridge University in just a few moments. But before we deep dive into that, sometimes we need to ensure reliable results with your immunology research, like all the time. From primary human stem cells to cell isolation kits, cell culture media, supplements, and antibodies, STEMCELL Technologies provides the tools you need for every step of your immunology research. Interested in cell isolation? Use EasyCep to isolate highly purified immune cells from virtually any sample source in as little as 8 minutes. Cells are viable, functional, and immediately ready for your downstream applications. Learn more at easycep.com.
Hi, everyone. Today we are talking to Dr. Francesco Colucci. He is a professor at the Department of Obstetrics and Gynecology at Cambridge University and a fellow of King's College. He will be talking to us, amongst other things, about his research exploring the role of uterine immune cells in pregnancy. And we're very happy to have you here. Thank you for joining us.
Thank you very much, Brenda. It's a pleasure to be here, and congratulations on your podcast, which is great.
So to dive in, I don't think of immunology and uteruses that much. I mean, I know I should. It's kind of an interesting immune privileged system. You obviously don't want your immune system going after a fetus, which is where I think of the immunology going in. But you really dive into some interesting like ILC related physiology, immunology that you've been able to uncover in the uterus. So could you kind of start there with at a high level what you've been able to find in uterine tissue as it relates to ILC function and specifically, and then fetal development?
Yes, thank you. So it's true what you say, that whenever we think about immunology of pregnancy, we think about this paradox of pregnancy. Why doesn't the mother's immune system why doesn't it reject the fetus, which is half mismatched? And that question has been really on the mind of immunologists of reproduction for decades, since Peter Medawar, who won the Nobel Prize in 1960 for his discoveries in immunological tolerance. He actually formulated that question. And we've been thinking in those terms for decades. But lately, we've really realized that actually the immune system of the mother engages in a conversation, a molecular conversation, with the wannabe placental cells from the fetus. So these are the trophoblasts, right? So I'm sure we'll talk about this later, but this sort of constructive interaction rather than conflicting tension between the immune system of an individual and another individual. So it all started when I was in Paris, actually, when I was a postdoc with Jim DeSanto, who is now one of the stars in the innate lymphoid cells world. And there was a person she was going to become a legend in the field of reproductive immunology. She is called Anne Croy. She's a retired professor. And we had just set up a model of NK cell deficiencies. And she was actually, commuting from Canada to Paris to use our mouse models to look at what would happen to reproduction in these mice who didn't have NK cells. This was a model of severe combined immunodeficiency. And she had such passion, and she basically influenced me and planted the seed. And then when I went to Cambridge, and I met another legend in the field, who is Ashley Moffitt, who is a sort of semi-retired professor here, and she was working with human uterine NK cells. Then I decided to start looking into mouse models of. Because we were quite strong in flow cytometry, like all self-respecting immunologists, we said, let's take a look, close look as a proper, immunologist looking at subsets. This is the mid 2000 sort of 2004, 2005. And you may remember that the first ever paper on innate lymphoid cells was published in 2008. And so in 2008, we also published a paper describing the phenotypic diversity of NK cells in the uterus. And some of them were innate lymphoid cells, but the word, the phrase hadn't been invented yet. So that's where we started. And then we realized that the NK cells in the uterus were not the same as the ones in the blood, which people had already seen in other tissues like the liver. And then little by little, we've learned that innate lymphoid cells are really part of the physiology of the tissue they belong to. This is true in the gut, in the skin, in the respiratory tract, and so is in the uterus.
So we know in the case of NK cells, and what you show in your latest work, that they have a very important role in mediating some of the restructuring of the trophoblast and the organization of the placenta that is dependent on NK cells. And I was actually quite surprised to hear, because when you think of NK cells, natural killer cells, if you always feel of NK cells killing MHC negative cells, or cell very important against viruses and such things. And I think it's hard to picture these cytokines from NK cells being so important for tissue remodeling. So maybe would you like to introduce us to particularly this part of uterine NK cells, and how then you studied their regulation in a way?
Yes. So that's a very good question. So we've got these cells that we like to call natural killers. It's a dramatic name, and then we find them in the uterus where they should help to build rather than to kill. And that's actually what they are. They are builders. This is when killers become builders is a phrase introduced by a colleague now retired called Le Boutier, who wrote a commentary paper on a paper published by Ofer Mandelbaum, a good colleague back in Jerusalem, who really showed that also human natural killer cells in the uterus help the vasculature to refurbish, basically, and the placenta to differentiate. Now, that may be shocking, but if we go back to our knowledge of natural killer cells from people's arms, so from healthy donors, you take the blood from healthy donors, and you look at NK cells, it's been known for many, many years that there are at least two types of NK cells, one of which is the true assassin cells. These are capable of killing tumor cells in petri dishes. And the other subset, which is 1 in 10 of these cells circulating in our blood, they're actually not so good at killing. They can, but they're not as good as the professional killers. And this other subset, this 1 in 10 blood NK cells, they are much better at producing cytokines. Now, if we go into tissues, and that does not have to be the uterus, if we go into the liver, if we go into any other tissue, we see that the majority of NK cells are actually the second type, so the type that produces cytokines. So there is already something inbuilt in the system which relates to what we were saying before, and that is innate lymphoid cells, whatever their lineage, type 1, type 2, type 3, they're really in sync with the physiology of the tissue. They've been influenced by the microenvironment there, and they influence themselves the microenvironment. So to go a bit more molecular and technical, a cytokine we find in abundance in the uterus is TGF-beta. And TGF-beta is found in the tumor microenvironment. So also in the tumor microenvironment, being a tissue, although abnormal, but also in the tumor microenvironment, unfortunately, there are more builders than killers within. So if you gate on natural killer cells, if you take them out of the tumor, you do a flow, and you ask them, are you the type that kills or the type that produces cytokines? Also in tumors, we find more types of cells which respond to the gating of NK cells, but they're actually better producing cytokines than killing. And that's perhaps a way of the microenvironment to corrupt the immune cells to help them. What do I mean? Well, we know that there are similarities between some NK cells in the womb, in the uterus, and NK cells in the tumor microenvironment, and that is they help the vasculature. And obviously, this is going to help tumor cells as well because it will bring in more oxygen. So that's an example of how the microenvironment influences the cells that are there in the tissue. So the majority of natural killer cells in the uterus are not killers. We can stimulate them to kill if we put them in vitro with the right cytokines, which again tells us that they are part of that lineage of killers, but they have adapted to the local environment and they are basically builders.
So to kind of go along this a little bit, you talk about a pathway that's responsible for the non-killer natural killer cells, which I'm going to call natural cuddler cells from now on, since they seem to be all happy and helpful. Natural cuddler, cuddle.
Oh, that's a great.
you can steal it. But for these non-killer cells, you talk about a pathway that's really interesting that I hadn't learned about before, and I was hoping you could explain a little bit, which is this CD94 NKG2A receptor, and then how it takes HLA-B packaged peptides, presents it to HLA-E, and then does something. Can you explain this like triple cascade with multiple HLAs, all in one non-killer cuddling T cell or killer NK cell thing?
Yeah, Jason, I can see your experience in industry and how you're good at making great names and funny names that people can remember. But we immunology academics, we are terrible at naming molecules and cells. So CD94 and KG2A, it's horrible. It's horrible. But let's call it A, just to make it simpler. So this is an inhibitory receptor, right? All respectable white blood cells, all respectable immune cells have inhibitory receptors, right? These are off switches that all immune cells have to sort of regulate the extension and duration of an immune response. And we're familiar with these now because we target them with immune checkpoint inhibitors in cancer patients. We reawaken the immune system goes into overdrive and kills the tumor cells. So these off switches, including this receptor, let's call it A, on NK cells, do the same things in NK cells. But in NK cells, they go to another level. And what they do, and this is the process that we've focused on, is they educate NK cells. Now, this seems something like a bit esoteric, but it has similarities with, and also fundamental differences with the education that happens to wannabe T cells that go into the thymus and that they are educated by interacting with self-MHC. Same for natural killer cells. They basically, if they have an inhibitory receptor for self-MHC, they are set up to spring into action when they are needed. Let's leave it there. I'm happy to answer other questions on the process if you want to know more. So this is the process of NK cell education. And this A receptor is on 95% of human NK cells in the womb, and about 50% of cells in the blood, and about 50% of cells in the womb of mice. So it's a relevant receptor. And we knew that education may be important for uterine NK cells to set them up for action. So we looked. So what does it do? This receptor interacts with self-MHC, but not the sort of famous HLA ABC that we know of, but non-classical HLA-E molecule. This is one of the most ancient HLA. And the receptor is also very ancient. It's been around since the time of dinosaurs, because it hasn't changed from mouse to human. So it has been in the last common ancestor of mice and humans 80 million years ago. All right. So this ancient receptor and this ancient MHC, if they interact, then they set up NK cells for action. So this is education. However, we people are divided roughly 50-50 in those who can express a lot of HLA-E and those who cannot. And that is determined by a peptide in HLA-B, which gives its leader peptide to HLA-E if I have methionine in position -21 of the leader peptide of HLA-B. But if my sister instead has inherited the threonine at position -21 of the leader peptide of HLA-B, she will have a poor HLA-E expression, and therefore I will educate my NK cells better than she will do.
That's complicated. I'm just going to put out there that we have a dinosaur era HLA that's getting another peptide from another HLA to map this out. And this was something. How is this determined? Like scientifically, like how did they figure out that you have like an HLA peptide handoff? Was this all GWAS studies and they found that through that initially, or do you know? Because like that is obscure.
I know who has found the importance of this dimorphism for NK cell education, and this is Amir Horowitz, a good colleague and friend now at Mount Sinai in New York, then at Stanford with Peter Parham, who is one of the giants of HLA and immunogenetics. Now, and it has been known for many years that HLA-E expression depends on other HLA peptides. But you caught me off guard there, and I don't have the answer for that. I don't remember who has discovered it and when.
That's fine, but it's interesting knowledge.
So we've used a mouse model of a knockout for this A inhibitor receptor and realized that all the things that the NK cells had to do in the womb were done in a sluggish way. These had consequences on placental, uterine, vascular remodeling in the uterus, placental gene expression and fetal growth, including brain development. And in humans, in a big GWAS study, we saw that women with that threonine that didn't allow a good HLA-E expression and therefore poor A education, NKG2A education, they had a greater relative risk of developing a disorder of pregnancy called preeclampsia, which also associates with fetal growth restriction often, and is supposed to be caused by insufficient vascular remodeling and placental differentiation. Yeah, it is. I realize it's complicated. I hope it makes sense.
What also it took me a couple of reads to understand, which I thought was also a little bit contradictory, is that actually the expression of this inhibitory receptor that is required for education of uterine NK cells is actually necessary for these cells to produce more interferon gamma upon when they find themselves in the uterus and they find trophoblast and they have to generate this placenta. So I guess this comes in the frame that you mentioned, that you either have cells that are cytotoxic or cells that are making cytokines, and they seem to be two different profiles. I think that really helped me understand. But I thought it was really fascinating how it shows how important it is, right, to for the evolution of all placental organisms to have these very basic cells that are always there, that are going to ensure the correct irrigation, the correct vasculature, and there's these innate cells that have been there for so many years, for so many evolutionary time. So I guess I wanted to ask you a little bit more about your career. So you are a physician scientist, so you're also a MD, and I guess that it must be very interesting to work in this kind of research that is so relevant for many women that, in this case, are suffering with this with these diseases such as eclampsia that are really hard to understand, and often we don't completely understand the source. You also throughout your study and your research, you moved in many different countries, and I was wondering, maybe you want to share a little bit about your experience and what that has taught you with our listeners that would be interested in a similar career path.
Yes, yeah. Thanks for the question, Brenda. Yeah, I've been around for a little while, mostly in Europe. So yes, I studied medicine, but I always had the back of my mind the idea that one day I would do research. I loved research, and so I finished medical school, and I had an opportunity, thanks to an organization called the Swedish Institute, to go to Sweden, to the University of Umeå, where I did my PhD, to simply do a research experience of 9 months. And I fell in love. That never stopped then, ever since. It was many years ago. And there I studied type 1 diabetes, which was also, very medically relevant, although I was using a mouse model. And then I remember, I don't know if this will be truly inspiring for younger colleagues, but I decided the town I wanted to do my PhD, and that was Paris. I decided I had to go to Paris. And I was lucky to start working with Jim DeSanto, as I said, who was just starting up his lab, and we were very successful, me as postdoc and he as PI. We started to work on development of NK cells and how they recognize and kill cancer cells. And then I moved on to have my first job at the Pasteur Institute as an associate professor. So as a member of staff at the Pasteur Institute, I had a permanent job. But then I had the good fortune so personal life got in the way. Although I love being in Paris, and my then-to-be wife also loved being in Paris, but her being British, we decided to give it a go to her native island. So we moved here in Cambridge about 17 years ago. And so when you change countries so many times, you see so many different academic institutions, you become more adaptable and you worry less about a defined career path. So whenever, students, either undergraduate students in college where I help them studying immunology medicine, or my PhD students in the lab, or even postdoc ask me for some help and advice in their career path, I always say, just do what you really are passionate about. And then a career path is best described backwards, so from where you have arrived, and you say what you've done, rather than trying to do always the right step, because it basically means that you're playing catching up and you're trying to fit and conform. While if you just, for example, I had an MD, I didn't have to have a PhD. I did it simply because I realized I was so bad in the lab, since I had no experience when I finished medical school at 27 or whatever, that I said, I really need to dig deep and spend time with the other PhD students, although they were, 5 years younger than me. In my case, it turned out to be okay. But what I like to say to younger colleagues is just focus on your personal growth and development, because if you focus on getting a Nature paper, you have one chance in 100 of succeeding. if you focus on your personal growth and development, you have 100% chance of succeeding. And in doing that, we will try to get the Nature paper anyway. And sometimes it does happen. But I think best investment is in yourself. It's what I say always to young people. And changing labs or countries, Brenda, you've done it as well. It is tough, but also very stimulating.
Couldn't agree more. Jason, you gotta move out.
Yeah. Well, no, I've done enough moving. I've moved a lot. But I guess my follow-up is it's about being an MD PhD. And so in the US, there's often a conflict for academic MD PhDs in that your institute wants you to be two people. Instead of being a hybrid MD PhD where you split, they want you to generate all the clinical revenue of a regular MD plus grant revenue of a PhD. And in so much as your grant revenue of a PhD goes up, then you can spend a little less time in the clinic, but the clinic always kind of comes up behind you and tries to suck you back in because clinical medicine in the US is so profitable to the institutions that there's this motivation to have that. Now, being in a place with something that, as far as I'm concerned, is a mythical unicorn, aka a national health system of some form, where there isn't a profit motive, do you guys have that conflict, or is because, or for other reasons, clinical medicine being treated not quite the same market-driven approach it is in the US, that you then don't have this conflict between the MD part and the PhD part of your life?
Okay, so Jason, I should clarify, I'm not clinically active, so I don't experience this conflict personally, but I do have other colleagues who do have that conflict. It is not as intense as in the US for the reasons that you just said, but there is, it is demanding, and there are structures in places for young colleagues who, so for example, the first author of the paper we just published in Immunity, Norman Shreve is called, is a young clinician who decided to do a PhD in our lab, and now he's a clinical lecturer, and he has some research protected time, but is also quite involved still with clinical activity. And so, yeah, there are challenges. It's very, very quite difficult. And, I could candidly say that I'm a failure in that I never managed to do both, because I would have loved to do both, but I was maybe in my sort of romantic approach to life and science. I said, I just have to dedicate myself completely to research, and one day I will go back to clinic. That has never happened and probably will never happen for this lifetime. So it is quite very, very difficult to do both. And I do admire people that managed to be so organized and so energetic to be clinically active and do an impact in science and research. I think it requires talent and skills.
I guess in that line of thought, we wanted to ask you, like to ask some questions, David, maybe not necessarily related to research, but of life in general of our guests. And I guess it's a good time to ask you, for example, what is the best piece of advice that you've ever been given? And doesn't need to be professional, also life advice that you'd like to share with our listeners.
Okay, thank you. So, so the best piece of advice, I have a couple, if I may elaborate. But the first one is from an uncle of mine, whose name was, unfortunately, passed away quite young. He was Tommaso Meo. He was also, like me, a clinically trained scientist, an immunologist, immunogeneticist, who trained with Ruggero Ceppellini, who's been one of the main names in the HLA. And so before going to Sweden for my first ever experience in research, I spent a few months in his lab. He had a lab at the Pasteur Institute. And I remember once he told me, as soon as you learn to do something with your right hand, I'm right-handed, challenge yourself to learn it, to do it with your left hand. And I went, what is he talking about? Why do I have to pipette with my left hand? And so, but then I realized, it's so important to keep challenging yourself, never get too comfortable, not just for the practical things, but also mentally. And I guess moving is part of that attitude. And then my PhD, one of my PhD supervisors, Takeshi Matsunaga, he also gave me a couple of very important advices, which I'm still working on. I don't think I've managed to do what he suggested me, and that was two things. One was too much information kills information. In presentation, in writing papers, I'm still struggling. I think I always overload in information, but that came with this sort of Japanese minimalistic, beautiful culture. And the other one was speak slow and think fast, which I also still trying to achieve. But I like to share these pieces of advice with my younger colleagues whenever they join the lab, whenever we have an opportunity. I think they've helped me, and they're still with me every day.
Thank you very much. They'll be with a couple thousand more people or more. Who knows? Once we go
Oh,
up.
wow.
Well, thank you for your time today. Do you have a lab website or a Twitter account or any open positions or anything else you want to plug here before we wrap up for the day?
No, I don't have open positions yet. We hope to have them in 2022, soon, a few months' time. I do have a Twitter account, and that is at Dr. as in Dr. Fran_Colucci.
And we'll be sure to tag that on our show notes as well for all of you. Thank you for being here.
Thank you for joining us. It was a
Thank
great
you very much. Again, congratulations for your podcast. It's a great success among all my younger colleagues, and we'll keep following you.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup papers. You can also reach out to us on Twitter at Immunopodcast or via email at info@immunologypodcast.com with feedback or to suggest guests if you can think of someone. See you next time.